Thank you so much. Thank you to the organizers for inviting me to give this talk. So, extracellular vesicles are now understood to be made by diverse mechanisms. And, this slide really shows how many different kinds and sizes of extracellular vesicles can be made. Exosomes are sort of one of the first EVs really described small vesicles between about 40 to 150 nanometers in size made within multivesicular bodies. Also, now we know that ectosomes are released from cells in a variety of shapes. So originally described as really large EVs. Small EVs the size of exosomes can be released from the plasma membrane of cells. And these are sometimes called microvesicles, but to avoid confusion, ectosomes is probably a better term because it really tells you it's coming from the plasma membrane. Apoptotic bodies, also one of the early described extracellular vesicles, but I think, for many years people really didn't think that they were something to be considered as a functional entity. I think it's something that we need to think about, that it's not just inert being released from cells, but there are a variety of shapes and sizes, and they could also have functional effects. And then there are a variety of additional vesicles that are sort of in panel C there that don't, necessarily fit into these boxes that I just described. For example, migrasomes, which sort of look like multivesicular bodies, but they're formed on retraction fibers being left behind cells. There are different ways to secrete vesicles, for example, from the amphisome that's shown there. That's a sort of combination body that's made from fusion of a multivesicular body with an autophagosome, and if it fuses with the plasma membrane, can be released. So what can I tell you about biogenesis of some of these different kinds of vesicles? I'm just going to talk today, because I have a very short period of time, about ectosome biogenesis, and spend a lot of time on exosome biogenesis. So ectosome biogenesis is diverse. So the one mechanism that we understand for how the larger vesicles are made is that the plasma membrane of your cell has a cortical actin shell underneath it, and if there's weakness or disassembly of the actin underneath the membrane, you can get a blebbing out of the membrane, and that combined with actomyosin contractility can pinch off those large vesicles. And I've also got R6 listed there because it can be an upstream signaling regulator of that process. In addition, there's flipping of lipids between the inner and outer leaflet of the membrane, and this is important for both the large EVs that are formed from the plasma membrane and also has been implicated in smaller EVs that are formed. So as I mentioned, small EVs the size of exosomes can also be released from the surface, and this can involve both lipid flipases to help induce membrane curvature, but also a machinery that I'm going to talk about in a minute, the escort machinery, is known to be recruited also to the plasma membrane in some cases, for example, by the ARRDC1 protein that's listed there, and can induce exosome-sized vesicles formation at the plasma membrane. So now I'm going to switch to exosomes. So exosomes are a type of small extracellular vesicle that are formed within endosomes in your cell. And so basically you can think of, receptor ligand complexes get endocytosed from your plasma membrane. They're in early endosomes, and then things get sorted out of your early endosomes. But some things, actually some cargos, for example, a classic cargo would be EGF, EGFR ligand receptor complexes, will get sorted into intraluminal vesicles. And these are future exosomes that will get secreted from the cell. And so as the endosome matures, it becomes a late endosome and a multivesicular body. And then that multivesicular body has multiple fates. It can fuse with autophagosomes or lysosomes and degrade the cargo, or it can dock with the plasma membrane and fuse and release those intraluminal vesicles that were formed as exosomes. And so there's a flux between these processes, and it can be shifted in one way or another to either secrete more or fewer exosomes. So I want to talk just a little bit about some mechanisms for molecular recruitment of cargo and formation of exosomes. So one principle is that cargo recruits the machinery that then bends the membrane and pinches off the vesicle. And so in panel A there is shown the most classic way of making an exosome, which is that receptor ligand complexes get ubiquitinated. That ubiquitin molecule recruits an escort 0 protein, which here in the oblong is listed as HRS STAM. That's an escort 0 complex. Escort stands for endosomal sorting complex required for transport. And so basically there's a series of escort molecules, and one recruits the other. So the escort, once it recognizes that cargo, the escort 0, it then recruits the escort 1 and 2 molecules, which recruit the escort 3. Escort 3 forms spirals around the membrane, which help both induce membrane curvature and pinch the vesicle off. So that's the classic mechanism that's known. However, there are, as we now appreciate, multiple ways to recruit escort at various steps in that process. So for example, in panel B is a mechanism Pascal Zimmermann's laboratory described for syndecan-syntenin complexes recruiting TSG101. TSG101 is an escort 1 protein and then recruiting the rest of the escort complex. Panel C shows a supposedly escort-independent mechanism of intraluminal vesicle formation. So this was reported first in 2008 and has been shown many times to be involved in intraluminal vesicle formation. So basically the way it works is sphingomyelin is a type of lipid found in lipid rafts. It can be acted on by neutral sphingomyelinase 2 to form ceramide. Ceramide has this wedge shape, and this is thought to induce membrane curvature, which is one of the first steps to making an intraluminal vesicle. Notably, tetraspanins, which are major EV markers, small EV markers, also have a wedge shape. It's not really known if that is involved in EV biogenesis or curvature, but it's something that people have been thinking about. Also, I should mention it's possible that late escort components could come in and pinch off that vesicle once ceramide induces the curvature. That's sort of an unknown or an open question, I would say, in the field right now. Okay, another aspect of exosome biogenesis or EV biogenesis I want to talk about is RNA binding protein complexes as a cargo. So if you look at the panel, I guess it would be on the left, that has the big multivesicular body. This is from a cartoon showing how different cargoes are recruited. So for escort, it's pretty clear how things get recruited. As I mentioned, ubiquitinated cargoes recruit escort. So it's sort of membrane-linked molecules. Also for tetraspanins and ceramide dependent, it's thought that cargoes that are embedded in lipid membrane domains are what's acted on they ride along as the membrane curvature happens and the ILV is formed. RNA and RNA binding proteins and other cytoplasmic cargoes, it's less clear how they connect to these machineries. I think that's still an open question. There was a paper from Jay Debnav's laboratory in 2020 that was very intriguing, suggesting that at least some of these could be recruited by interaction with the autophagy protein LC3 via LC3 interaction motifs. My lab has done some work on this showing that biogenesis of RNA-containing EVs takes place at ER membrane contact sites where there are RNA granules. However, I think it's really still an open question and unclear how the RNA machinery connects to the machinery that makes interluminal vesicles. Okay, another point that I want to make is once you make these interluminal vesicles, the MVB has to go to the cell surface and dock and fuse to release those interluminal vesicles. So we know some of the players involved in that. For example, Rab27A and B are involved in the trafficking and docking of MVBs with the plasma membrane, and then SNARE proteins like synaptoglycan 7 and others are involved in the fusion with the plasma membrane. So I'm going to show sort of a practical example of where we've done some work that I hope will illustrate some of these concepts I just talked about. So one of the things that we've wanted to do in our work, because we are interested in understanding how exosomes promote cell migration, is to actually visualize exosome secretion at a subcellular level. So we designed a way to do this based on a fluorescent, a pH-sensitive green fluorescent protein that we tagged to an exosome marker CD63. So CD63 is a tetraspanin. Tetraspanins are highly enriched in small EVs. And this one happens to be typically localized to late endosomal lysosomal organelles in the cell, especially multivesicular bodies. Late endosomes have an acidic pH, so the fluorin pH-sensitive GFP tag is quenched at acidic pH, but it fluoresces at neutral pH once the multivesicular body fuses with the plasma membrane. So we first made and published this construct in 2015, and just taking some wide field movies initially, we discovered that we saw the fluorin CD63 not only at the plasma membrane, but also in exosome trails that were being left behind the migrating cells. And there's a zoom of those exosome trails in the upper right. Using total internal reflection fluorescence microscopy, we could see that there was a burst of fluorescence before new adhesions formed. So the red is the adhesions and the green is the fluorescence. So if you look at the arrows, you'll see a flash of fluorescence and then a red adhesion forms. We made a brighter version of this construct in 2020 and used it to show that cells migrate on paths of deposited exosomes. And so how can we determine that these green things that we're seeing in the movies are really secreted exosomes? So we've taken a couple of ways to do that. Oh, and this is just showing that using this pathfinding index, cells are really migrating along the paths of deposited exosomes. So one thing is, if you do correlative light electron microscopy, you can see that the green fluorescent trails correspond to small EVs and retraction fibers. And if you hone in, you can see that the small EVs are actually green, and a larger EV, which is 700 nanometers, is not green. So the CD63 is marking small EVs. Also, we used a couple of escort proteins to colocalize with the CD63, TSG101, which is escort 1 protein, ALEX, which is an escort accessory protein, and found that indeed the CD63 colocalized with those proteins, with ALEX better than TSG101. And then I told you that Rabs could promote multivesicular bodies. So if you inhibit Rab27A, which promotes MVB docking with the plasma membrane, it actually leads to less deposition of this fluorescent CD63 outside the cell. So in this short time, I hope I've given you a flavor of how extracellular vesicles are made. So just to summarize, EV biogenesis involves cargo recruitment, induction of membrane curvature, and vesicle scission. There are multiple variations on how each of those are achieved, and we know less for large EVs, especially in terms of how cargoes are recruited for large EVs. Exosome secretion involves multivesicular body trafficking, docking, and fusion with the plasma membrane. It can be tracked with pH-sensitive live imaging reporters, and each step is subject to regulation. So thank you for your attention.